Welcome to the lecture series in food microbiology. So far in this series, we saw which are the common microbes which predominate different kinds of food, what are the conditions for their existence and proliferation in different types of food. We talked about the extrinsic factors, we talked about the intrinsic factors. Then we saw what are the practices which we have to adopt to prevent their growth and to have an extended shelf life for the different kinds of food. We also saw how do we diagnose or how we detect the pathogens which are present in different kinds of food. Another important aspect of food microbiology is find out the causative organisms of foodborne diseases. Whether you are talking from the point of view of a food industry person, a consumer, or from the perspective on the other side, how to diagnose, that you have a lab which diagnoses or which tests the food products, identification of the different microbes and the other organisms which can spoil food as well as which can cause diseases to the people who consume them is equally important. The causes of foodborne gastrointestinal disorders can be broadly divided into three groups. One, those disorders which are caused from consumption of food and water containing viable pathogenic microorganisms or their preformed toxins. So, now in this category we are talking about food or water contaminated with live organisms or the metabolites, the toxins which are formed by them. So, the food would be full of that. The second category is from the ingestion of pathogenic algae, parasites and their preformed toxins. The third category is for reasons other than the viable pathogens or the toxins. Ingestion of toxins naturally present in many foods. So, toxins can be present even if there is no live organism. If the toxins are present in the food, if the food is not sufficiently processed, that is whether cooking, preservation, whichever is the technology we are using, if that is not processed, many of these toxins would still be stable and would be able to react with the human intestine, causing gastrointestinal disorders. And this many of the common food like mushrooms, peanuts, fruits, vegetables and many seafoods, all of them have toxins which are present. Most of these toxins get destroyed during our normal cooking methodology. But if it is not proper or if you have practicing improper hygiene or improper cooking practices, then these toxins would still be present in the food and would be liable to cause gastrointestinal disorders on people who consume it. Toxins can be formed in some food. For examples are the biological amines like histamines that are formed in fish, cheeses and fermented meat products. The presence of toxic chemicals in contaminated food and water like heavy metals and some pesticides. This would also cause gastrointestinal upsets to people who consume them. Then you have allergy to some normal components of a food. For example, this picture shows rashes formed because of consumption of food containing gluten, bread, biscuits, anything with wheat, anything related to wheat which contains gluten. So, some individuals are allergic to the gluten in the cereals and develop digestive disorders following the consumption of food containing gluten. So, for that you will see on the skin, it can be gastrointestinal disorders, but more than that the allergy would first manifest as a rash or a scratch or an itching or what you call as hives which will be present on the skin. This picture shows the person who is allergic to peanut. See the pink colored rashes on the face and the swollen portion in the mouth parts. Many people, so when you are allergic to peanut, even if your food product contains minute amount of a paste or any product related to peanut, it will immediately trigger allergic reactions on you. So, that is where the labeling of the food becomes very important, saying that this product contains this taken from peanut, or this product contains this, contains gluten. So that composition becomes important, and it tells the people that if you are allergic to this, you cannot consume this particular product. Genetic inability to metabolize normal food components. Some people do not contain certain enzymes which are required to catabolize certain components. For example, lactose intolerance. In lactose intolerant people, they do not secrete the enzyme lactase. They do not have the enzyme lactase in the small intestine. Because of this lack, they will not be able to consume any product which contains lactose. So, milk and milk products would have to be avoided. It will give them digestive disorders. Then you have the nutritional disorders like rickets, which is caused by calcium deficiency. And the last type of intestinal disorder would be indigestion from overeating or for any other reason. The federal regulatory agencies of the US define foodborne disease as an outbreak when 2 or more people become sick with a similar illness or symptom from the consumption of the same food from the same source, and the epidemiological investigations implicate either directly or indirectly the same food from the same source as the cause of the illness. So, how do you say there is a foodborne disorder outbreak? So, if you know 3 or 4 people or even 2 or more people who had the same food from the same place, a restaurant, and both of them show similar symptom within the same number of hours after the consumption of the food, and also later on when the investigators try to find out the source, the cause is related to the same source either directly or indirectly. So, then you say that there is an outbreak of a foodborne disease. However, in the case of botulism, because of the high fatality rate, even when only one person has the illness, it is considered an outbreak. Normal gastrointestinal disorders, we implicate them as a foodborne outbreak only if 3 or 4 or more number of people fall ill after consumption of the food, but not in the case of botulism, which is a lethal toxin. Botulinous toxin is a lethal toxin. It is almost instantaneous and it causes death. It is very severe. So, even if one person after consumption of a food dies or has symptoms of botulus poisoning, then it is considered to be an epidemic, to be an outbreak. And for chemical poisoning too, even a single case is considered as an outbreak because chemical poisonings are also more hazardous. Foodborne diseases in humans result from the consumption of either food and water contaminated with viable pathogenic bacterial cells, or spores as in the case of infant botulism, or food containing toxins produced by toxigenic bacteria and molds. So, humans have to either consume the food and the water which have been contaminated with viable live pathogens, the cells, or bacterial spores. If it is a gram-positive spore former, there, remember, spores live long, they are dormant. Even if the viable cells are not present, the spores, because of their nature, will continue to exist. They are difficult to destroy also. So, even if the spore is present in the food, when the conditions are conducive for the spore, it can germinate to give rise to the live organism, which in turn is pathogenic. So, even if a spore is present, then also you can have a foodborne disease, or you can also have toxins if the food contains toxins produced by the bacterium or the mold, or in the case with toxigenic algae, there is an outbreak of a foodborne disease. On the basis of the mode of illnesses, these can be divided into 3 groups: intoxication or poisoning, infection, and toxico-infection. So, how does this disease manifest? Based on that, we divide it into 3 types: intoxication or food poisoning, foodborne infection, and foodborne toxico-infection. In intoxication, the illness occurs as a consequence of ingesting a preformed bacterial toxin or a mold toxin because of its growth on food. So, either a bacterial toxin or a fungal toxin is present in the food and you consume the food having it, as a result of which you have an illness. So this is a foodborne intoxication. A toxin has to be present in the contaminated food. So once the microorganisms have grown and produced the toxin in the food, there is no need for the viable cells during the consumption of the food. So you don't even have to detect the debris of the live organism. It is enough if you can detect the toxin which is present in the food concerned. A typical example is staphylococcal food poisoning or the staph food poisoning, a foodborne infection. Here, illness occurs as a result of the consumption of food and water contaminated with enteropathogenic bacteria and virus. In this case, live organisms are present in the food. You consume a food containing the live organisms, you ingest it, because of which you fall sick. That is a foodborne infection. Here it is necessary that the cells of the enteropathogenic bacteria and the virus remain alive in the food or the water during consumption. Viable cells, even if present in very small numbers, have the potential to establish and multiply in the digestive tract to cause the illness. So even if very less number of these live cells are present, once inside the gut environment, once you have consumed it, they can divide, they can replicate and then establish themselves, then cause the disease. So a typical example is salmonellosis, hepatitis A, the disease called hepatitis A, etc. These are caused by a foodborne infection. Toxico-infection. Here, illness occurs from ingesting a large number of viable cells of some pathogenic bacteria through contaminated food and water. In toxico-infection, whatever you consume, the cells have to be large in number, not small. It has to be very large in number. And generally, these bacterial cells either sporulate or die and release the toxins. So once you consume this large amount of bacteria, they would either be spore formers or once inside the gut they will die. But during this process they will secrete toxins. Since the initial consumed load of organisms itself is very high, the toxins produced are also going to be equally high in amount, creating an adverse reaction inside the person who consumed them. Typical example is Clostridium perfringens gastroenteritis. In addition to the pathogenic microorganisms associated with foodborne illnesses, some bacterial species and strains normally considered to be non-pathogenic can cause gastroenteritis, especially in susceptible individuals. These we call as opportunistic pathogens. Human gut has a microbiome which are the natural microbiome. These include LAB, then you have the bifidobacterium, you have the E. coli. A large number of gut microbiome organisms are present as your normal flora and fauna in the gut microbiome. But many of these can also become opportunistic pathogens. A typical example is E. coli is your normal gut dweller, but you do have a pathogenic state of E. coli. So when you happen to consume food containing that pathogenic E. coli, once you consume the live cell, it can, because of its tendency, it can transform your normal non-pathogenic bacteria into this, thereby asking it to produce the endotoxins, making you more ill. So then your normal gut dwelling E. coli becomes an opportunistic pathogen. They are normally required to be alive and present in large numbers when consumed through contaminated food. Other than your normal gut microbiome, you have the other normal non-pathogenic organisms also. So if your food gets contaminated with it and you happen to consume it when it is present in a large number in the food matter, once it gets into you, it can become as a pathogen causing illness. So these are opportunistic pathogens. Now, how do we classify? So you need the pathogen to be present. The pathogen has to contaminate the food concerned. Then, how is the food preserved? The preservation, the temperature, storage, everything has to be conducive for the pathogen to grow. So once the pathogen has grown to a sufficient level, and it can either cause intoxication, it can cause a bacterial infection, or it can go for toxico-infection. Now, for intoxication, we have to consume the food containing the toxin. For bacterial infection, you have to consume the food containing the live cells. For toxico-infection, you have to consume the food cells containing a large number of bacteria. Now, if it is for viral infections, then it has to be confined, then consumption of the food. Viral pathogen has to be present in the food and you have to consume it. So, this is the normal sequence of how a foodborne disease manifests itself on a person who consumes the contaminated food. Now, how does a pathogen survive in the intestine? The pathogen has to face several hurdles, several obstacles to be an intestinal pathogen and to cause an illness. First of all, it has to survive the passage through the extremely acidic environment of the stomach. Your stomach has dilute HCl, so it's an acidic environment. The pathogen, once consumed live, has to live and exist and survive the acidic environment of the stomach. Some pathogens are aided in this process by the protective effect of the food, and some survive the acidity by the use of their adaptive acid tolerant mechanisms. If the pathogen is engulfed or entrapped within the food particle, that itself will give it the ability to survive in the acidic environment. But in some cases, they do change, adapt themselves to survive in the acidic environment. Then it needs to attach to or to colonize the intestinal walls in such a way that it can increase in numbers. It needs to go find a foothold, grab a foothold, stay there, get attached, then start dividing, because increased numbers is required for manifesting as an illness. The mucous layer that covers the intestinal mucosa is regarded as being the first line of defense encountered by the enteric pathogens. So, are you able to find a foothold and or grab an intestinal cell and get a foothold, or don't you have any attachment, any places where you can attach yourself? If you can have an attachment site, then you can manifest as an infection, proliferate. If you don't have an attachment site, then you cannot hold, you will just get washed off. So, that particular, so the mucosa, the lining actually is involved in this. Certain intestinal pathogens grab hold of to the receptors which are present on the epithelial cells, attach to themselves, then start their activity. So, such receptors have to be present on the epithelial cells for the intestinal microbes to attach themselves and start proliferating. In the case of Listeria monocytogenes, it overcomes the mucus barrier by removing the mucus through the aid of Listeriolysin O. So, the mucus layer will prevent the pathogen from grabbing a foothold. What if you remove the mucus? That is what Listeria does. It removes the mucus by secreting a substance which is called as Listeriolysin O, otherwise called as LLO. Once the mucus lining is dissolved, then the receptor would be present. Some pathogens like the pathogens, like Clostridium perfringens, it does not has to attach to the intestinal tissues. They have the capacity to defend themselves against the host defense mechanism like the gut associated lymphoid tissue. Any organism has a gut associated, like your normal immune system, there is a gut associated lymphoid tissue which maintains the immunity inside the gut. So, the pathogen which is trying to get a foothold via food has to have the capacity to withstand, to defend itself against such host defense, and it has to be able to compete with the large heterogeneous microbiota of the gut. As I said, human gut microbiome is huge. So, any organism, any pathogenic organism which you consume and goes to the stomach and the intestine would be competing for the carbon source, for the energy source, for the ATP source with those intestinal dwellers. So, for the pathogen to proliferate, to get a foothold and proliferate, it should be able to compete with those normal intestinal dwellers, take over them, in the sense that it should be virulent enough to survive in conditions where the microbiota, the normal gut microbiota is huge. The pathogen is going to be very less in number. Even in such small numbers, it should be able to get a foothold and start dividing. This is the gist of the competitive exclusion, in that the harmless microbiota, once attached to all available sites on the intestinal walls, will exclude pathogens. So, your normal gut microbiota, they also have a suitable receptor site on the intestinal walls. If all the receptor sites are blocked by the normal gut microbiota, then the pathogen will not have any receptor. So, competitive exclusion happens, the pathogen will get washed off, it will not be able to proliferate because all the sites are blocked by the harmless normal gut dweller. The gastrointestinal tract has a low oxygen environment where the predominant organisms are anaerobes. But it has also been shown that in the case of Salmonella typhimurium, this growth in a less oxygenated environment actually induces in it the ability to enter the mammalian cells. So, that which earlier was did not enter a cell, now because of its growth in the low oxygen environment is able to enter a mammalian cell and survive. Once attached, the organism needs to be able to either elaborate toxic products, for example, the Vibrio cholerae 01, or cross the epithelial wall and enter phagocytosic or somatic cells, for example, Listeria monocytogenesis. So, once the organism has got attached, it would either have to start producing the metabolites, toxins, or it has to cross the epithelial wall and enter the phagocytes or the somatic cells, then start its activity. It cannot continue by holding on to the receptor, getting attached and remaining there. Some foodborne pathogens use at least two strategies to survive in the low pH conditions. One is the RPOS gene. This is the normal, this is the same. This includes for the alternate sigma factor called as sigma 38. A sigma factor is a subunit of the bacterial RNA polymerase that varies in size, and it has been reflected by the superscript in kilodalton as delta 38. Sigma 38 regulates at least 30 proteins. The RPOS is regarded as being a sigma factor that will specifically affect the stationary phase events in some enteric pathogen. Especially this is seen in E. coli, Yersinia enterocolitica, Shigella, and it allows them to survive at pH 2.5 for over 2 hours. So, the RPOS gene will activate a certain set of genes downstream from it and ask the organism to secrete certain metabolites, which in turn will allow this organism to survive and start proliferation at acidic environment where the pH is less than 2.5 for more than 2 hours. It is responsible for the induction of specific sets of genes such as those that control the glycogen synthesis, thermotolerance, hydrogen peroxide resistance, starvation survival, and extreme alkaline resistance, osmotic stress, etc. And in the case of some salmonella, the expression of genes on the virulence plasmid also SPV. So, the activation of RPO here in turn will activate the virulence plasmid SPV. Together, these two will start in overproliferation of the organism concerned, making the person more sick. Acid resistance, defined as the percentage of the cells that survive exposure to pH 2.5 for 2 hours, is well studied in Shigella. After these organisms leave the colon, they enter the stationary phase outside the host, and upon ingestion by another host, they are already acid resistant and low numbers can survive through the acidity of the stomach. So, suppose a person consumes a food with Shigella, it has gone into the intestine. Because of the presence of the RPOS gene and the other factors, slowly within 2 hours, it becomes resistant and it can be now survive in the acidic environment. Okay. So, now this becomes a stationary phase. Outside suppose this acid resistant Shigella goes out with the fecus outside. There it will remain outside, it will now remain in a stationary phase. But suppose this again goes back into another human host because of contaminated food and water. Remember, this organism is already acid resistant. So, the moment it enters another human intestine, it would be able to start proliferation, be able to survive because it has already become acid resistant. Even then, the low numbers, even if it has not become completely acid resistant, even in low numbers, they can still survive through the acidity of the stomach concern. Foodborne intoxication. Food poisoning of microbial origin occurs by ingesting a food containing a preformed toxin. Some general characteristics of food poisoning are: 1. The toxin is produced by the pathogen while growing in the food. The pathogen proliferates in the food and does its metabolism. During this metabolism, the toxin is produced as a byproduct, and this toxin is now present in the food. This toxin can either be sensitive to heat, it can break down when heated. So, during cooking, the high heat maintained can kill the toxin concerned so that the food becomes safe, or it can be heat stable. Ingestion of a food containing active toxin, not viable microbial cells, is necessary for food poisoning, except for infant botulism, in which viable spores need to be ingested. Symptoms generally occur quickly, as early as 30 minutes after ingestion, and symptoms differ with the type of toxin. Enterotoxins produce gastric symptoms, neurotoxins produce neurological symptoms. Febrile symptoms are not present. So, in the case of a toxico-infection, a food intoxication or a foodborne infection, the organism or its toxin or any other category has to be consumed. It has to withstand the adverse environment in the gut, in the intestine, survive. If it is a live organism, it has to proliferate. If it is a toxin, it has to survive the cooking temperature, the processing temperature, that is the heat stability, and it is also has to be stable in the acidic pH of the gut. Then it can cause the foodborne illness concerned. In this session, we can have two or three questions. One is, how do we classify the organisms? Is it based on the symptoms which they produce on people who consume the contaminated food? 2. How does processing technologies help in controlling foodborne infections? Thank you.